So this whole unit, this whole section of this unit today is going to be about how we regulate the expression of these genes. Okay? And we're going to learn about how this occurs in prokaryotes and in eukaryotes. Now, it's really important that we're able to take certain genes in our DNA and turn them on or turn them off. Because what this allows is for cells to become specialized, right? What I mean by that is, if you look at your body, every single one of the cells in your body has the same DNA. But the cells in your liver, the cells in your pancreas, right? They're made up of the same DNA, but the proteins that they are making are going to be different, right? Because they have different specialties. They have different jobs. So what that means is that if we take our DNA, we're not going to express all of it. We're only going to express the parts of the DNA that are relevant to accomplish the job of that cell. So the pancreas will only activate certain genes to produce proteins that it needs. The liver will only activate only certain genes to produce those proteins. So that allows us to now specialize in these cells. So in order to do that, we have to be able to control whether our genes are turned on or whether the gene is turned off. So we're going to start with the simpler example, which is in bacteria. Now, if you think about bacteria, they're unicellular, right? So they don't have that cell specialization like we do, right? But bacteria must also be able to regulate the genes that they're expressing to some degree. So in bacteria, in prokaryotes, we're going to call this group of genes that we're studying an operon. So operons are a group of genes that can be turned on or off, and they're made up of 3 parts. Promoters, we know that word already, right? We learned that word in transcription. That's where transcription begins. This is where the RNA polymerase binds. Do prokaryotes need transcription factors for this? No, only eukaryotes need transcription factors, right? The RNA polymerase can bind directly to the promoter. We're adding another part now. We have something called an operator. This is the on and off switch for the gene that we care about. And then we have the genes. These are the genes that make the enzymes, that code for those enzymes. All right. We have 2 types of operons. We have repressible or inducible. Don't try to memorize these 2 words. Just think through them logically. A repressible operon means I'm able to repress it. So do you think this operon would normally be on or off if I'm able to repress it?
On.
It's normally on. So a repressible operon is normally on, but I can turn it off, or the bacteria can turn it off. Inducible would be the opposite, right? I can induce it. So normally it's off, but it can be induced to turn on. Mm-hmm. All right. Now we have another gene called a regulatory gene that's going to produce something called a repressor protein. This repressor is going to always be expressed. So we're making this repressor, and it's always floating around. But whether that repressor is active or not will depend on whether certain molecules are present in that solution. Okay? So in order to understand how these repressor proteins work, we need to flash back a little to our allosteric interactions. Remember that when we learned about proteins, right? So remember we talked about allosteric activators. When we have these substrates binding to the allosteric site, that's not the active site. And in this case, we are going from an inactive to an active form. So this is an allosteric activator. Then we have allosteric inhibitors, where we have an active protein, but we can inhibit it by binding this to the allosteric site. The active site changes, and this no longer functions. Okay? So we're going to be doing this to that repressor protein. So let's see how that works here. we're going to walk through how repressible and inducible operons work with two specific examples. You have to be familiar with these. The first is the trip operon. The trip operon in bacteria regulates the synthesis of a protein called tryptophan. Tryptophan is a protein that bacteria need a lot of. So that's why it's a repressible one, because normally it's on. It's constantly making tryptophan because the bacteria needs it. But sometimes if there's too much tryptophan, it doesn't want to make it anymore. So it's going to repress it. Let's see how it does that. Okay, this kind of walks you through a description of that, but let me just walk you through it with this diagram here. Let's look at it normally. So normally we have our promoter. RNA polymerase binds to the promoter. It reads down, and these are some different genes that are all related to the production of tryptophan. So the end goal here is this binds to the promoter, which allows us to produce tryptophan. Great. Now let's say that there is a bunch of tryptophan floating around. The cell doesn't really need it right now. What happens? Notice how we have the repressor here? And you see how the repressor is not doing anything. Right now, it is in an inactive state. But when the repressor binds to tryptophan, tryptophan activates the repressor because it binds to its allosteric site. Now, when the repressor is active, it will go to that new region we learned about called the operator, and it binds to it. If we bind here, notice what happens to the RNA polymerase. When it connects to the promoter, it cannot get past this repressor. So it cannot reach these genes over here, and no tryptophan will be made. Now, we wait a little bit of time. No more tryptophan is made, but the cell is using that tryptophan, using the tryptophan that's accumulated. So this tryptophan that's attached here will eventually get used up. If this gets used up, what happens to the repressor? It detaches. And if it detaches, then we can go ahead and continue transcription, allowing us to make more tryptophan. You see how this is kind of like a form of negative feedback, where if too much tryptophan is made, we stop more from being made. This is called a repressible operon. Yeah.
So does a repressor only work so is a repressor only activated when the tryptophan gets binds to, and then it'll bind to the operator? Is a repressor always kind of bonded to the operator?
No. If the repressor is attached to the operator, then no transcription would happen. But in this example, when there's no tryptophan, the repressor does not attach to the operator. This brings us to an inducible operon. The specific example that you need to know is called the lac operon. Now, the lac operon controls the synthesis of lactase, which is an enzyme that breaks down lactose. Is anyone here lactose intolerant? No. So if you're lactose intolerant, you struggle to make the enzyme lactase, so you can't break it down. Now, again, I'm going to talk about this, just walks you through it. I'm going to walk through this with the diagram, but this is what lactase does. It takes lactose, which is a disaccharide, a 2-ring sugar, and it breaks it apart into glucose, right? Monosaccharides, which now the bacteria can use as an energy source. This is available as energy, this is not. So in order to get energy, the bacteria needs to make lactase to break this down and give itself glucose. So what this means, though, is normally the cell does not want to make lactase. Because think about the bacteria. Will the bacteria be surrounded by lactose all the time? Probably not. It's a changing food source. So why would I keep making lactase, the enzyme, if there's no lactose available for me to break down? That's a waste of energy. So we have a regulatory mechanism where normally a repressor is attached to the operator and no transcription can occur. You see how this is active, even though there's nothing attached to it here, right? But when allolactose, for your purposes, you may want to make a note of this so you don't get confused. Allolactose and lactose, you can think of them as almost synonymous. They're like slightly different forms of each other, but when you have lactose, you also have allolactose mixed in. So all this is saying is when there is lactose present, what does it do? It binds to the repressor, and notice how that removes the repressor. You see how we're inhibiting this repressor now? When we remove that repressor, transcription can occur, which will allow us to produce all the proteins we need to break down lactose. Now, this is called an inducible operator because we will only turn it on when lactose is present. When there's no lactose present, it's shut off. And that's makes it very efficient. Okay. Do we have any questions on how this works? All right. This slide here goes a little bit more in depth, showing you how there's some different ways that it regulates as well in other scenarios. For our purposes, we're not going to dive into this, but if you're curious about it, feel free to ask me about it. You will not be assessed on this, so you might want to make a little note to yourself so you don't panic when you're studying. Okay, let's see how much of that you actually retain today. So trying not to look at your notes, take a minute. Can you compare and contrast lac versus trip operons? Okay, what's the similarity between both of these operons? Any similarities?
They have a repressor.
Okay, they both are using repressors. Yeah, that's good. They're both regulating transcription, right, of a gene, just in different ways, right? Key differences between these, hopefully you talked about. One is repressible, right? The trip is repressible, and the lac is inducible. So which of these two things do you think the cell needs to make more of? Tryptophan or lactase? Tryptophan, right? All right. And what are the three parts of an operon? See if you remember that. We start with the promoter region, where the RNA polymerase binds. Then we have the operator region, where the repressor will bind. That's the on-off switch. And then you have the genes of interest that you're making. Okay. We're almost there. Now we're halfway done. We now need to move into eukaryotic regulation. Now, unfortunately, we're not going to go as deep in the eukaryotic pathway, but it's a lot more broad. Because for eukaryotes, we need to specialize ourselves. So we need a lot more levels of control. Not just, do I turn the gene on or off? That will be one layer. But we now need to add other layers to this way of regulating gene expression in eukaryotes. So we call this differential gene expression, just saying that we express genes differently, right, in different cells. That allows us to specialize them. This will kind of show us how we have many different levels in which we can control and regulate the expression of a gene. So we're going to start way up here at the beginning, talking about our chromatin, the DNA itself. So one thing is the structure of the chromatin. The Biozone mentioned this a little bit in one of the earlier sections, but it talked about the winding of the DNA around those histone proteins. Now, if the DNA is really tightened or really loose, that will impact the accessibility of those genes by the proteins that we need, right? Remember, we need RNA polymerase and we need transcription factors to get into that DNA. So one way we can regulate which genes are accessible is by either doing DNA methylation, which tightens the DNA, tightens the chromatin, or histone acetylation, which will loosen up that DNA, that chromatin. So histone acetylation makes it more accessible. So those genes are easily reached by RNA polymerase and transcription factors. But DNA methylation will do the opposite. It'll tighten it up so those proteins can't get in. So we cannot express those genes. Right? So if my liver cell only needs a certain section, we might only keep that section loose. The rest of it will keep tightened up. This leads to a pretty interesting phenomenon called epigenetic inheritance, where these chromatin modifications, anytime you see the word chromatin modification, it's talking about the methylation and the acetylation. Okay? These modifications are not changing the DNA. The sequence of the DNA stays the same. But what's interesting is these modifications can actually be inherited, right? Part of heritable to future generations. And this explains why when you have twins, their DNA is identical. But it is possible that one twin may inherit a disease while the other one does not. Because one twin might have inherited some of these modifications that was made by the parent. Usually these modifications are wiped clean when you have a child, but sometimes they're preserved. And that can lead to some discrepancies in twins. So this phenomenon is called epigenetic inheritance. Okay, we've talked about how we regulate it at the chromatin level. Now let's look at the transcription level, like in bacteria. This is the equivalent of the bacterion we just learned. We have another name here. We have a region of DNA called these control elements. And these are sections of non-coding DNA that will serve as binding sites for activators and repressors that will either increase transcription or decrease transcription of particular genes. So let's look at a picture here. Here we see we've got our promoter and we've got the gene of interest. Now in order for us to do transcription, remember, we need two things. We need a RNA polymerase and we need various transcription factors to attach. There are certain regions of DNA, in this case, notice it's upstream of it, called control elements. In this case, we have something called an enhancer. This is promoting transcription by helping the transcription factors and the RNA polymerase bind to the promoter. They're stabilizing that structure. This will enhance transcription. So here's another picture of that. Notice here, we've got an activator attached to this site. This activator will help transcription factors and the RNA polymerase to bind to the promoter. But instead, what if I attach a repressor to this region of DNA that's upstream of the promoter? If I bind a repressor there, it's going to stop RNA polymerase and transcription factors from binding. That will stop transcription. So essentially all we're saying here is whether you have an activator or a repressor present, that will either promote transcription or inhibit transcription. So now we're controlling transcription at the level of do you transcribe or not? Okay, now let's say transcription happens. We make it. Okay, we did the transcription. Now we need to process that RNA. What are the ways that we process it again?
5' cap.
5' cap.
Poly A tail.
3' poly A tail, and
Intron splicing.
we splice. But is the way that we splice the same every time? No, remember we can cut them out differently. So here's another level of regulation. How am I going to cut this? Because if I cut it differently, it will result in a different protein. You see how that's controlling what we're expressing. So alternative splicing is going to be another level of regulation. Great, now I've made my mRNA. Whew, we're getting there. Now we need to go do translation. Okay? We have another layer of regulation here. Because translation can be activated or repressed by these initiation factors. For example, two you should know are called microRNAs and small interfering RNAs. What these can do is they can either break the mRNA apart, degrade it, or they can stop it from binding to the mRNA, to the ribosome. So here we are preventing translation from happening. We're controlling that. So again, another layer of control. I made the mRNA, but am I going to let it get translated or not? Okay, and we're going to leave it at that. So, to recap, you can regulate eukaryotic gene expression by changing the chromatin, by turning transcription on or off, by alternative splicing, and by do you allow it to translate or not due to these microRNAs and small interfering RNAs. All right. Yes.
Can you like stop it from leaving the nucleus? Is that another level?
I'm not sure if there's a very specific one that will just like stop it from leaving the nucleus. Perhaps I'd have to see whether that's like a specific stage there that's getting inhibited. All right, here are some just basic words to be familiar with. Don't worry too much about the details of these next couple slides here. Just understand that in eukaryotes, remember I talked about specialization in cells? When you're growing, you start out with these things called stem cells. Stem cells can differentiate into any type of cell, right? But then they're going to specialize, and at that point you're going to regulate which genes are turned on or off. And again, all of this process of building your physical structures, this is called morphogenesis. Like physically, genesis, the creation of, right? Creating your physical things. How do the cells know what to turn into? Right? So how does a cell know, oh, hey, my job is to turn into a liver cell? Well, in the egg, they have these chemicals called cytoplasmic determinants and this concept of induction, where cells signal each other. Again, don't worry too much about this. Just understand that there are substances that will trigger the cell to specialize and turn into target cells that the body needs. Okay? And lastly, again, don't worry too much about that, is apoptosis. A lot of the cells, to finalize the structure, you need to do apoptosis. The classic example is your hands and your feet, right? You have webbed hands and webbed feet, but we do apoptosis to get our more dexterous fingers. All right, so that is the regulation of gene expression. We did it, yes?
Like stem cells can turn into like anything. It's not even like an embryo. Why is stem cell research so controversial?
Because people then argue when has life started? When is this embryo alive? Right? So by removing those stem cells, are we destroying the potential for future life? Right? Is that ethical? Is that not? So it really depends on people's definition of when is something considered alive, I guess, at what point. And so there's a lot of debate about that. Yeah. Okay, last bit here. Let's watch this little video about your genes are not your fate.
Is this the woman with prion disease?
One way to change our genes is to make new ones, as Craig Venter has so elegantly shown. Another is to change our lifestyles. And what we're learning is how powerful and dynamic these changes can be, that you don't have to wait very long to see the benefits. When you eat healthier, manage stress, exercise, and love more, your brain actually gets more blood flow and more oxygen. But more than that, your brain gets measurably bigger. Things that were thought impossible just a few years ago can actually be measured now. This was figured out by Robin Williams a few years before the rest of us. Now, there's some things that you can do to make your brain grow new brain cells. Some of my favorite things like chocolate and tea, blueberries, alcohol in moderation, stress management, and cannabinoids found in marijuana. I'm just the messenger. What were we just talking about? And other things that can make it worse. It can cause you to lose brain cells, the usual suspects like saturated fat and sugar, nicotine, opiates, cocaine, too much alcohol, and chronic stress. Your skin gets more blood flow when you change your lifestyle, so you age less quickly, your skin doesn't wrinkle as much, your heart gets more blood flow. We've shown that you can actually reverse heart disease, that these clogged arteries that you see in the upper left, after only a year become measurably less clogged. And the cardiac PET scan shown in the lower left, the blue means no blood flow, a year later orange and white is maximal blood flow. We've shown you may be able to stop or reverse the progression of early prostate cancer by extension breast cancer simply by making these changes. We found that tumor growth in vitro was inhibited 70% in the group that made these changes, whereas only 9% in the comparison group. These differences were highly significant. Even your sexual organs get more blood flow, so you increase sexual potency. One of the most effective anti-smoking ads was done by the Department of Health Services, showing that nicotine, which constricts your arteries, can cause a heart attack or a stroke, but it also causes impotence. Half of guys who smoke are impotent. How sexy is that? And we're also about to publish a study, the first study showing you can change gene expression in men with prostate cancer. This is what's called a heat map, and the different colors, and along the side on the right are different genes. And we found that over 500 genes were favorably changed. And in fact, turning on the good genes, the disease-preventing genes, turning off the disease-promoting genes. And so these findings, I think, are really very powerful. They're giving many people new hope and new choices. And companies like Navigenics and DNA Direct and 23andMe that are giving you your genetic profiles are giving some people a sense of, gosh, well, what can I do about it? Well, our genes are not our fate, and if we make these changes, they're a predisposition, but if we make bigger changes than we might have made otherwise, we can actually change how our genes are expressed. Thank you.
So we can see here that, yeah, a lot of the things in your life are due to your genetics, yes. But not all of it is fixed in stone, right? A lot of the gene expression is controlled by the environment, right? Your diet, your stress levels, all that, right? So which genes are expressed can be influenced by your lifestyle as well. So a couple things to think about. Okay, so that's gonna be it for gene expression and regulation. We're going to do a little activity now where you're going to do a simulation to learn about how operons work in the lac operon specifically in prokaryotes. Okay, so that's found in the Google Classroom.